Hi, everyone. I'd like to thank the organizers so much for inviting me to be a part of your conference, and I really wish I could be there in person. So I'm happy to talk about my lab's work to create designer biofilms on a chip and other types of living material. So in general, my lab is interested to use bacteria to produce materials. So can we replace traditional chemical synthesis approaches with a biological approach that's more sustainable, more simple, more inexpensive? But one big question arises, if you want your bacterially produced materials to have a shape, how could you spatially pattern the materials being produced? So to address this, here's the solution that my lab came up with. Here are some students that I coached for the iGEM team from TU Delft in 2015, who invented the first 3D printers that print bacteria. So we're holding in the middle of this picture the printer that the students developed. It's the cheapest bioprinter that I know of ever developed. It's so cheap because everything is made with Connect toys that are easy to assemble yourself, from the ground up. The fact that our printer is colorful and made from toys makes it a really engaging educational tool. So anyone's welcome to go to our paper and download directions for how to make your own toy 3D printer. So I really love the idea of 3D printing with bacteria. So I've been continuing on, even after this iGEM project was over, to continue developing this new technology. So here's what our current version of the 3D printer looks like. So let's look at how it works. So this printer, as far as we know, is the second cheapest 3D bioprinter ever developed. It's much more precise than our toy printer was. How it works is that the ink, it's bio ink, it's a liquid bacteria culture. It's in a syringe in the syringe pump. And the syringe pump basically squeezes the syringe plunger so that the ink is extruded through some tubing over to the print head where it's deposited onto the surface. So we have full automated control over how fast the bio ink is being extruded, as well as where the print head is over time, because it's a commercial 3D printer that we just bought off the shelf. So when we extrude our bio ink, we're extruding liquid culture onto an agar plate. But how do we get our liquid culture to form a long-lasting three-dimensional shape? So here's the chemistry that we use. So we're printing onto an agar plate that contains not only food for the bacteria, but also some calcium ions, such that when we deposit our bio ink, it's liquid bacteria culture, but it also contains alginate. So alginate has fantastic properties because after printing, the calcium in the agar will diffuse up to interact with the alginate in the bio ink, which causes it to polymerize. So now our liquid bio ink is a solid, so it's a hydrogel. The bacteria are trapped inside this polymer matrix. But the great thing about the fact that it's a hydrogel means that it's mostly water still at this point, even though it holds its shape very well, so that food can continue to diffuse in and feed the bacteria. Inducers can come in and diffuse in to induce specific production of engineered chemicals or products from the bacteria. And if you want to print a second or a third layer on top of the first layer, the calcium will continue to diffuse up, and you can make many layers, at least up to 20 layers, on your three-dimensionally printed structure. So with our 3D printer, we can print a variety of arbitrary shapes, either 2 or 3-dimensionally, as long as there's no internal cavities, right? And we have, depending on our printing speed and the chemistry that we use, we can get down to sub-millimeter resolution. So a single stripe can be printed with 0.2, 0.3 millimeters in width. So we get nice, reproducible 3D printing of bacteria with high resolution. So what kind of materials do we want to make with our bacteria? So the first application that we targeted was, can we print models of biofilms? So biofilms are a very common way for bacteria to live in nature. So they're when bacteria don't live individually, but they live a community lifestyle held together by spatially structured extracellular matrix components. Biofilms are incredibly important to human society. They're often used beneficially for purifying water or extracting, for example, copper from mine tailings. They can also cause the majority of infectious diseases, as well as dental plaque that causes cavities in your teeth. So, their large importance to human society makes it really important that we can have good reproducible model systems to work with biofilms in the laboratory. However, my students that developed 3D printers talked with various industries, for example, toothpaste industries, toothpaste making companies that work with model biofilms, and they find that from week to week, the biofilms they make in the lab aren't all the same as each other, and they can't reproduce the multi-species nature of natural biofilms. So we thought that our 3D printer could be a great way to make really reproducible biofilms that were the same every week, that would also be able to capture some of the spatially patterned multi-species aspects of natural biofilms. So our approach is we print bacteria that have been engineered to produce curline, which is the major protein extracellular component of biofilms for many species of bacteria. It's what sticks the bacteria together to each other and down to the surface on these biofilms. So after printing, they're printed onto a surface containing inducer, which diffuses up to the bacteria, induces them to make the curly protein, which is then exported outside of the cell, where it will spontaneously assemble to make these long curly fibers that stick everything together. So what happens when you do this? So when we print these engineered biofilms, do they then behave like natural biofilms? So one thing we wanted to look at was, do they have the characteristic low oxygen concentration, where the oxygen's been depleted and not replenished due to the thick extracellular matrix? To look at this, we printed 4-layer biofilms, either natural biofilms as like a positive control, or engineered bacteria either that express or don't express the curly fibers. After we let them mature and grow for a week, we test the concentration of soluble oxygen at different depths using a microelectrode. So for this here, we're looking at different biofilm depths on the x-axis and the oxygen concentration. We can see that comparing the gray and the orange curves, we see that when we go to the orange and we add the curly production, we see for the first time really sharp decreases in oxygen concentration and a long, section of depth of the biofilm that has no detectable oxygen. And we actually see even lower oxygen than in our natural biofilm strain in blue. So we have really good reproduction, reproducing of the oxygen depletion phenotype of these natural biofilms. Also, if you want to have our biofilms behave like natural biofilms, you need to have emergent resistance to antibacterial treatment. So natural biofilms, the matrix is so thick and hydrophobic typically that many antibacterial compounds can't penetrate biofilms, and they're no longer able to remove the bacteria like you would want them to during medical treatment. So to test this, again, my postdoc, Shrikant Balasubramanian, printed these 3 different types of biofilms, let them mature, and we cut out the same volume of each sample treated with an antibacterial compound, in this case, ethanol, and look to see how many bacteria survived. So here we're looking at, we see like increasing concentrations of ethanol, and the y-axis shows colony forming units, how many bacteria survive the treatment. So we see, as there's more and more concentrated ethanol, we see fewer and fewer bacteria survive. But you can see that in orange, the engineered bacteria that produce the curly fibers survive at a much higher rate than in gray, the bacteria that don't produce the curly fibers. So our engineered bacteria in orange can show emergent resistance to ethanol, very similar to what we see in blue for the natural biofilm strain. Let's see. So 3D bacteria printing, I think, is a really interesting application for lots of different things. So I think it's great for making standardized biofilms for developing treatments to cure people that are infected with bacteria. It's an interesting way to make reusable devices for purifying water, for example, or extracting compounds from the environment in a way that the bacteria is really robust and it's easy to transfer it from vessel to vessel without losing your biomass. I think it's an interesting application. I'm currently joining a new consortium to work on, can we 3D print bacteria over top of plastic before it's deployed in the environment for degrading it over time so that we don't have plastic building up in the environment and leaving plastic waste. Of course, since we can print bacteria, we don't only have to print biofilm bacteria, we can print bacteria that produce biomaterials. So can we make other materials my lab makes? So can we print bacteria that my lab uses that make graphene conductive materials or Nager, so biomineralized tough structural materials in any shape that you want since we've 3D printed them? Can we print, my lab shows that you can print photosynthetic microbes like microalgae that might act as artificial leaves, so photosynthetic materials that can fix carbon or fix solar energy in the form of sugars. Could you print, as an idea from the student lab, could you print bacteria that makes cellulose in the shape of your certain body part that had a burn or a lesion and make a personalized skin graft that fits to your personal shape? I think that's a really interesting direction as well. And of course, bacteria and organisms in general are great at responding to the environment. So we take advantage of this by printing materials that contain bacteria that sense environmental cues and respond by healing the material or changing the toughness, the stiffness of the material, because they detect contaminants in the environment and then sequester or remove them. Could you make, there's this idea from some labs that you could make even kind of like living bacteria that kind of act like tattoos on your body that would sense your personal physiology and respond by changing color to alert you of, do you need certain medications at that moment, for example. I'm going to zoom to this last part, but here's just a few examples of the fact that we can print microalgae. So we can print bacteria, we can print microbes in materials that change color over time in a way that they're physically robust, grow on their own just by having sunlight as their energy source, and you can share and distribute them, which I think is a really interesting application. So thanks for listening to my talk. My lab works not only on engineered biofilms, but also we use bacteria to make structural materials, conductive graphene materials, optical materials, and space materials. And I'm really excited about this new direction of using synthetic biology to make materials in a way that's really making high quality, desirable materials that's sustainable and easy to make and inexpensive. So I'd like to thank everyone in my lab that helped work on these projects over the years, and everyone's welcome to please send me an email or get in contact if you're interested to know more or if you have any questions. So thanks so much. I'll end the recording.